Hi, this is a video about light reactions, that part of the photosynthesis process that is dependent on light. This is Kim Foglia's PowerPoint from Explore Biology, narrated by Dr. Meade. We are continuing in the very same PowerPoint as the photosynthesis overviews video. So we're talking about the light-dependent reactions, and that is an electron transport chain. You learned about one type of electron transport chain in cellular respiration. So it's going to be similar, but we're talking about completely different proteins in the process. So the difference between the mitochondrion electron transport chain and the chloroplast electron transport chain is the kind of proteins that are in there. We still have electron acceptors like NADPH, which is, of course, money in the bank because it's carrying electrons that are high energy. And another similarity is that we're creating a proton gradient across the inner membrane. And what do we use a proton gradient for? Well, as you can see in this picture here that we've seen a whole bunch of times, that we drive the process of ATP synthase by having a proton gradient. Okay, so we're going to make some ATP using a proton gradient. Right, so you remember, okay, that it had to take place, okay, in a plasma membrane. So where's the plasma membrane that we're going to be using here? Well, it's the thylakoid membrane, okay, which is a lipid bilayer, just like the other kinds of membranes. And so the similarity here that you're seeing is no accident, of course. Remember from endosymbiotic theory that we are concluding that organelles were originally some prokaryotes, and that one prokaryote, absorbed other prokaryotes, and those evolved to be organelles that functioned, within cells. So we can assume and conclude that mitochondria and chloroplasts have a similar ancestor. Right, so we're going to have that ATP synthase involved also. So this is just to remind you that these are similar processes, still calling it electron transport chain because it's still moving along high energy electrons, but it's going to have different proteins involved. But the one that's consistent is ATP synthase. Remember how I said that you could find ATP synthase in a bunch of kinds of membranes. And so we're definitely going to see that same enzyme in the thylakoid membrane that we saw in the mitochondrial membrane. So, okay, there's our chloroplast. There's our buildup of protons in the thylakoid membrane. And it's going to drive the process to make the ATP. See, now I shut off those sound effects and I just want to make my own all the time. I don't know which is worse. You decide. Annoying Dr. Meade sound effects or annoying PowerPoint sound effects. I'll go with mine. I like mine. Okay. So here is the electron transport chain of cellular respiration, just to kind of remind you so we can sort of show you the kind of why we kind of notice that respiration is sort of the opposite of photosynthesis. Okay, so we're we're using up the NADH, okay, and we're getting some ATP. Oxygen combines with protons to form water, the hydrogen ions, okay, and electrons are being handed off from one electron carrier to another, okay, using the proteins, and then these little proteins, the cytochrome proteins and the quinone lipids, okay, which is not a protein. So now, that's the mitochondria, okay, the transfer of chemical energy from food into ATP using the electron carrier NADH. So how would we we run this in reverse? Okay, if photosynthesis and cell respiration are sort of the opposite of each other, but we're still talking about electron transport chain, how would this kind of look going backwards? Okay, oh yeah, so here we're generating water. So, so we're, all in the process of breaking down sugars, moving electrons along, generating water as a byproduct. So what if we went backwards? So here's the electron transport chain of photosynthesis, and this is that picture that I said, kind of like in chapter 9, that we keep coming back to orient us. All right, and we're talking about light-dependent reactions of photosynthesis happening in the thylakoid membrane, the little pancake stack there. Okay, so how is this the opposite? Well, we've got a couple places where light comes in, and photosynthesis is endergonic. The electron transport chain is being driven by light energy. And we're going to look in more detail about how this occurs in the next slide. But this is to give you the kind of big picture. So if we're kind of looking at it as the reverse of the electron transport chain and respiration, okay, here we're gaining NADPH, an electron carrier, okay, and we're gaining some ATP, and those, okay, are going to go on to the Calvin cycle where we make sugar, right? So what's coming in is light and water, right? What's coming out is NADPH and a hydrogen ion and some ATP. So that's the big picture about what's happening here. Water going in, light going in, NADPH and hydrogen ion and ATP out. Okay? And and don't forget about our friend ATP synthase, making ATP down there because of the proton concentration gradient. And to orient you again, that we're looking at a cross section of the thylakoid here. So here's the thylakoid membrane, the lipid bilayer right here. Then we have the thylakoid space and the stroma, which is like the cytosol of the chloroplast. So the proton gradient that's being set up here is the higher concentration of protons in the thylakoid space, leading to them wanting to move out into the stroma, and that's powering our ATP. Okay, we're going to go into this in more detail on the next slide. Sorry, I just took a big long pause, so I lost my train of thought. Alright, so you're probably wondering where the carbon dioxide comes in, because we didn't talk about carbon dioxide going into this situation. Well, we're not going to talk about using carbon dioxide until the light independent reaction, the Calvin cycle. So this is where we're using water, this is where we're using light, and we're ending up with NADPH, which is a lot like NADH, it's just another electron carrier, okay, with a little extra phosphate thrown in there. I probably should have pointed that out to you earlier, but don't be intimidated. It's the same kind of thing. It's just another electron carrier, and we make an ATP. So the thing that I want you to notice here is that light is going into these two photosystems, and photosystems are here. It goes into 2 first, but the reason they're numbered 1 and 2 is because number 1 was discovered first. They didn't know that there was this other one, so it's messed us all up forever because it's confusing to say 2 before 1, but there you have it. Science is like that sometimes because it's, you know, when you discover something and you name it, it's a pretty heavy-duty thing. You discover something, you decide, do I name it after me? Do I name it after somebody else? Do I name it what it is? And even then it can get messed up. So just to reiterate the main thing that's happening here, okay, this is our electron transport chain in the thylakoid membrane. Light comes in, okay, and we're going to look at the electrons that come out of this. We are going to split water, okay, because as little particles get excited in here, a photosystem is like a combination of a bunch of different kinds of molecules. There's a bunch of proteins in there. The chlorophyll is in there. All right, so the light is going to excite the chlorophyll to create an excited electron, and that creates a little space here that pulls the electron out of water. So we're left with oxygen gas as a byproduct. So that's where we give off our oxygen, and oxygen is non-polar, so it can pass right through the membranes and go out as a gas through the stomata. All right, and also from the water we get a couple protons, hydrogen ions. Okay, and so the electrons get pulled along here. We get a little bit more proton action there. We get another bolt of light, some photons in here, exciting electrons, moving along. This becomes our electron carrier. All right. So the whole point of this is converting light energy, okay, into a chemical with a whole bunch of energy to be used. Okay. So we are using light. We are using the water. We are creating a proton gradient so that we can have a little ATP. We are creating an electron carrier to carry electrons, and these two products are going on to the Calvin cycle, and oxygen is being released. Okay, so with the electron transport chain and respiration, remember that our hydrogen ion, our protein concentration was going from outside the membrane to in, but here the, kind of showing us the perfect backwards reaction, that we have protons on the inside creating a gradient, and they're going to move to the stroma, and ATP synthase spins along and makes the ATP. That's the big picture. Okay, chloroplasts transform light energy into chemical energy of ATP and use an electron carrier called NADPH. And we generate our oxygen. All right, so, oops, I gotta shut my sound effects back off here. There we go. All right, so photosynthesis, of course, we're using sunlight. Okay, respiration, we're using sugar to drive that process, but we're still moving electrons around to run the ATP synthase pump, so that the pump is going to pump protons from one side to the other of the membrane, building up a proton gradient, drives the flow of protons through the ATP synthase and bonds that phosphate group hanging out there to the ADP, so that we get ATP. And ATP, powerhouse. Yeah? Okay, we know that. That's a review. Right. Oh, just to remind you that this is kind of showing again how this enzyme showing up in multiple membranes kind of reminds us about the role of evolution, that there's a common ancestor at work here between mitochondrion and chloroplast. All right, so let's talk about the pigments. All right, so in the thylakoid, right, in the chloroplast, we have a stack of thylakoids, and in one thylakoid, okay, here's our thylakoid membrane. You see the lipid bilayer there. All right, and then sitting here are a cluster of pigment molecules embedded in the membrane. All right, so here's what chlorophyll looks like. It has a porphin ring. Okay, this is the part of it that does the light, and then it has a hydrocarbon tail. So these lines are representing each of the carbons, okay, and you would have a hydrogen all along there. So they just didn't put all those hydrogens in. So that's the structure. Do you have to memorize the structure? No. You have to know that chlorophyll, though, has two main components, a porphyrin ring and a hydrocarbon tail. Chlorophyll and other pigments are embedded in the thylakoid membrane. Okay, because chlorophyll is not the only pigment. We know obviously that you know, green trees have other pigments in them because in the fall, when we're not seeing so much chlorophyll, we get to see all those other beautiful pigments coming to light, the carotenoids, the oranges and yellows, right? And there are two chlorophylls, chlorophyll A and chlorophyll B, that differ in this group right here. It's a, oh, I'm sorry, I can't read it and I don't remember. CH2 maybe, and then a CHO in chlorophyll B. Your picture in your book will have it. Yep, CHO in chlorophyll B and oh boy, my eyes. CH3 in chlorophyll A. Right? So they are arranged in what we call a photosystem, a collection of molecules. Let me show you a molecular picture of a photosystem. So I'm going to pause for a second. All right, so here's a picture from Wikipedia of photosystem 1. All right, so it's not just one type of molecule in there. There's a whole bunch of them being held together. You see a lot of different proteins, and all this green back here that you see is the chlorophyll. But of course, you can have other pigments as well. So it's called a system because it's a mass of different kind of molecules. Okay. So there is a structure and function relationship between how everything's arranged in a photosystem. And our penguin is going to ask us, how does this molecular structure fit its function? And they want to, at this point, just know about this chlorophyll molecule. How do you think that the structure is related to function? Why don't you think about that for a second and see if the class can kind of discuss and come up with an answer to that. All right, so if you kind of remember just sort of thinking about other things that have tails, okay, this is a hydrophobic tail, so it anchors each chlorophyll molecule into the position there. And in each photosystem, we have a lot of chlorophyll molecules, probably hundreds, I would guess. And this ring is kind of the perfect shape to receive light energy. Okay, it's actually a related molecule to hemoglobin. Hemoglobin, of course, is carrying oxygen around, but it's receiving that oxygen and able to carry it. Here we're receiving light and able to transmit. We have a bunch of high energy electrons that are just ready to give up their electrons here, and then this nice hydrophobic tail to anchor it into place. All right, so you may or may not be familiar with the light spectrum, that there's some light that's visible light, visible to us, of course. We usually show it in this direction from short wavelength to long wavelength. The shorter the wavelength, the higher the energy. The longer wavelengths have lower energy. And down here in the shorter end, we have UV rays, X-rays, and gamma rays. And over on the longer side, infrared, microwaves, and radio waves. All right, my dogs are crying for dinner. I gotta pause. Okay, we're continuing with our look at light. Okay, here it comes. Those are all our colors in the light spectrum. Okay, violet, indigo, blue, green, yellow, orange, and red. Why are we talking about light right in the middle of this? Well, of course, the plants are absorbing light, and we want to talk about what parts of the spectrum they are absorbing. Okay, so here we go. So photosynthesis gets energy by absorbing wavelengths of light, and it's not absorbing every wavelength of light, okay? And the different pigments are absorbing and reflecting different wavelengths of light. So chlorophyll A absorbs best in the red and blue wavelengths and the least in green. So that is why we see it as green, because it's absorbing the red and blue and reflecting green. And the other pigments, accessory pigments that have slightly different molecular structures, absorb light of different wavelengths. So here you can see chlorophyll A, okay, which is the big one, okay, chlorophyll B, and the carotenoids. All right, and so chlorophyll A and B are reflecting back sort of different shades of green. So chlorophyll A tends to be the blue-green reflection, chlorophyll B more of the yellow-green reflections, and then the carotenoids are absorbing down here and appearing yellow and orange. All right, so we're seeing what they do not absorb. So what we're seeing are these colors in here that they don't absorb. These are the colors that they like to absorb and use. Right? So they're absorbing every color except the one that they're reflecting. Okay, so those are the other pigments that are also in those photosystems, just to point out. Okay, so all those pigments are sitting in the photosystems, and they absorb different wavelengths of light and have slightly different functions along the way in the plants. So why are plants green? Well, they're green because the main chlorophyll pigments are absorbing here and reflecting here. So they are really happy absorbing red and blue and reflecting green, whereas the carotenoids, the yellow and orange pigments, are absorbing here, okay, and reflecting out in here. Make sense? I hope so. This is kind of an easy concept now that you see it, right? Oh, I'm always ahead of the penguin. All right, photosystems. Phew, let's talk about them. We've got two of them in the thylakoid membrane, and they are a collection of chlorophyll molecules. Of course, we already talked about that, and they act as light-gathering molecules. And the first one we're going to talk about is photosystem 2, because it was discovered second. It's got chlorophyll A, right, and P680, meaning absorbing 680 nanometers wavelengths of red light. And photosystem 1 has chlorophyll B, and it's P700, absorbing 700 nanometers wavelength of red light. So a slightly different part of the spectrum. Right, so the P is for photosystem. So these two photosystems just work a little bit differently. 680 nanometers versus 700 nanometers. So these are the two most important pigments that we're going to talk about. This is showing you the thylakoid, how light comes in, gets chlorophyll excited, and then we start hopping around some electrons. So this is a series of transfer of energy until we hit this primary electron acceptor, which is the reaction center for chlorophyll. Okay, so this is considered the reaction center, and then we have electron transfer. Okay, so there's the oops, there it goes, reaction center. So photons are absorbed by clusters of pigment molecules, and they're like little antennae, like antenna sticking up, right, in the thylakoid membrane. So when any antenna molecule absorbs a photon, it's transmitted from molecule to molecule until it reaches a chlorophyll A. Okay, and that is the reaction center. At the reaction center, is the primary electron acceptor, which removes an excited electron from the reaction center, chlorophyll A. That begins the light reaction. So the two photosystems don't compete with each other. They work synergistically using the different wavelengths of light. So just to reiterate, okay, that this is a cluster of pigment molecules in a photosystem. Okay, we're inside the thylakoid. Okay, there's a photosystem with a bunch of pigments. The pigments are sitting in the photosystem. Remember the picture of the photosystem with all different kinds of proteins and then a bunch of that green chlorophyll sitting there. Right, so the pigment molecules are like little antennae ready to take on a photon of light. So when any antenna absorbs a photon of light, it transmits it from molecule to molecule until it reaches a chlorophyll A. Okay, and then that is the reaction center. So when a photon hits the chlorophyll A, gets chlorophyll super excited to remove an electron, and once we have an excited electron, we can begin the light reaction. Good so far? So that's what this picture is showing you. We're seeing a photosystem. Photosystem has a bunch of pigments with little antennae ready to receive a photon. It transfers the energy of the photon until it hits a chlorophyll A. Because not every one of these molecules is a chlorophyll A, and we can't really move on until we've got a chlorophyll A with an excited electron ready to go on to the electron transfer chain. Right, so let's get into that electron transport chain a little bit more here. Okay, so we're looking at that picture that we saw before, but we're kind of putting it into context with now what we know about pigments and photosystems. So we have two places where light comes in, the two photosystems, and photosynthesis is endergonic, so the electron transport chain is driven by light energy. All right, so there's photosystem 2. Okay, with having chlorophyll A. Remember, we have that photosystem with a whole bunch of antennas sticking up, transferring around the energy until it finds a chlorophyll A to get an excited electron going. And there's a photosystem 1 that has light going in, but it wants a little bit of a different wavelength of light, and we're going to do that transfer of energy until we find chlorophyll B. Okay, so here's photosystem 2, which comes first. Okay, here's light coming in, energy of the photon being transferred around until it hits that reaction center with the chlorophyll A, and there's that P680, 680 nanometers of light, and 2 electrons are given off. And this is showing us the energy of electrons. Okay, so photosystem 2, as I said, it's that P680, 680 nanometers of light, and chlorophyll A. Okay, so there's step 1. So light comes in, okay, electrons come out. Right, once we hit the chlorophyll, chlorophyll gets excited, puts out a couple of electrons. All right, and the electron passes from the chlorophyll to the primary electron acceptor. So we need to replace an electron in chlorophyll, right? Because now chlorophyll has given up an electron. Okay, so let me back up for a second to show you where the 2 electrons are coming from. We hit the chlorophyll, chlorophyll gets excited and gives up an electron. Then we have an empty space in chlorophyll and we need an electron to replace it. So an enzyme takes the electrons from water. Remember, this is where we're splitting water. Okay, we take an electron out of water and give it to chlorophyll. And when we do that, okay, that reaction is going to split water into two hydrogen ions and an oxygen ion, which combines with another oxygen ion to form oxygen gas, O2. And the O2 is released to the atmosphere. Okay, so there we go. All right. Oops, why is it going again? Okay, there we go. There's our splitting of water, releasing our oxygen, and it's gonna give off two electrons, one of which is replacing the missing electron of chlorophyll A, and one is our other electron represented here. Okay, so the 1 and 2 is a little awkward here, but all right. So there's our water splitting. Okay, oxygen and two little hydrogens. Right, that becomes oxygen gas and gets diffused out of the cell. All right, and we get to breathe it. And here's our hydrogen, right? And we're going to get, when we are in this process of splitting water, these hydrogens aren't going to hang out with each other. They're going to give us two electrons and two protons. Okay, so I'm going to go through this whole thing over again because it's super confusing, right? All right, so light comes in, hits an antenna in the one of those pigment antennas. Energy gets transferred around, hits the chlorophyll A, okay? Chlorophyll gets super excited, gives off an electron. Now, chlorophyll is in need of an electron, and that's a powerful need. A need that's so powerful, it splits water. When we split water, now over here, all right, remember there's this pull. Okay, so the pull of the need of this electron in chlorophyll splits water. And we're going to end up with the oxygen being diffused out and water, or the hydrogen ions that are left, splitting, where we have two protons. Remember, we're going to need some protons because we're trying to create a concentration gradient down the road, and two electrons. One of these electrons is going to be incorporated into chlorophyll, and one of these electrons is going to go here, so that we have two electrons going to the primary acceptor. Phew! All right. If you want to see the similar image, it's on page 190, and you know, one of the things your book points out is that this P680 is one of the P680 electrons, rather the P680 is like one of the strongest biological oxidizing agents that is known. The electron hole has to be filled. So that's what I was saying, that, you know, that after that electron is given off, the need to replace it is huge. So you can imagine the power that's required to split water. All right. So remember that, you know, we're talking about that first part of the photosynthesis equation. So we're not talking about the CO2 yet. We're going to talk about that with the Calvin cycle. But right now we're talking about the light component and the water component. So why does a plant need water to live? Okay, it needs water because it wants water's electrons and protons. And remember that hydrogen is the most abundant element in the universe for a reason, because it can so easily, you know, give us protons and electrons. I mean, it's number one. So these hydrogen ions, aka protons, are going to be so important, and these electrons are also so important. So this is the essence of life right here, of why do we need, why do plants need water? This is it. This is why plants need water. Okay, and it needs water because it needs its water's electron, and it needs those protons. So what do you have to know here? Well, the first step in this electron transport chain, okay, number one, it's happening at photosystem 2. Photosystem 2 is P680, 680 nanometers of light. Okay, photosystem 2 has chlorophyll A. Okay, so I forgot what I said. If I was numbering these things to have you write them down, I got myself confused for a second. So I'm going to reiterate. So number one, we're talking about photosystem 2. It's P680. It's chlorophyll A here. Number two, this is where light comes in. It hits an antenna pigment, and that energy is transferred until it reaches the chlorophyll A. Number three, when it reaches chlorophyll A gets excited and gives off an electron. Number four, the missing electron in that molecule at the P680 is so strong that it splits water. And lastly, when water splits, okay, it replaces that electron, and another electron goes to the primary acceptor, and protons move on to the next step. Okay, oxygen gas is diffused out of the cell. Okay, or not necessarily out of the cell, it can be used for cell respiration if you're a plant. Okay, it can also be excess oxygen given out through the stomata. Okay, so there's our chloroplast, the thylakoid membrane. We are going to have a buildup of protons. When we have that buildup of the proton gradient, it drives the ATP synthase to make ATP. Okay. All right, so now, all right, we did all this part. We've got our primary acceptor, PQ, the cytochrome complex, and PC. Okay, and the electron flow provides energy for chemoosmotic synthesis of ATP that we just talked about. So this is the electron transport chain part, and electrons are moving from a higher energy state to a lower energy state, and when that happens, we get to give off some ATP. All right, and we're going to take the electrons over to the, well, you're going to watch the transition right here. So there's the two electrons we talked about in the previous slide. All right, there they are. Okay, going down the electron transport chain, and we have the movement of the protons. Okay, creating our ATP with our ATP synthase. So this is step 4 and 5 in your book version on page 190, but here it's 3 and 4. That was orso breaking. Shh, buddy. Shush. Lay down. Okay, so this is the intermediary step before the next photosystem. Okay, there we go. All right, and that ATP is going over to the Calvin cycle so that we have the energy to make our carbohydrates. And so the whole point of this is to get energy. The whole point is to transition this energy of the sun into the chemical energy of carbohydrates. And the part of that process that we're doing now, okay, is getting a little bit of ATP out of this whole idea of getting electrons out of the sun. All right, so we are still, just to give you a, relate it to the book, we are still in the non-cyclic energy flow here. All right, so we need a second photon. Okay, this is where photosystem 1 comes in, right? The P700, right? The 700 nanometer chlorophyll B. So similarly, we have a bunch of pigments in here with little antennae. All right, we need a second photon, a shot of light, to excite another electron back to a high energy state. So the second electron transfer chain drives the reduction of NADP to NADPH. So let's make this happen. So in comes the sun, okay, finds its way, same deal, using the antenna of the pigments, to the P700, the chlorophyll B, and gets it super excited so that electron is given off. Okay, got 2 electrons. All right. All right, and the electron that came down here is going to be the one that fills the vacancy. Remember that strong Meade that's pulling the electrons to fill the vacancy. But because we were giving off electrons with this chlorophyll, we're still going to end up with 2 electrons going off. Okay. And, oops, I didn't want to move out of there so quick. There we go. So I want to reiterate the entire thing right now. I know this is super confusing, so hang in there, and this is a long video, so we'll probably have to chop this up into another part. So, firstly, light comes into photosystem 2, finds its way to chlorophyll A, gets it excited, it gives off an electron. The need for an electron in this molecule is so strong, it splits water. So one electron fills the spot, and the other one goes on here to the primary acceptor. So now 2 electrons go down a chain, providing the protons necessary to drive a little formation of ATP. Now some more light comes in to photosystem 1, finds itself to chlorophyll B. Chlorophyll B gets excited and gives up an electron. Now it's got a hole, it's desperate for an electron to fill. It takes one of these electrons, and the other electron from here goes on out, so we've got 2 electrons moving onward. Okay, so now we're using those electrons, going down a little chain, so that we can turn NAD plus into NADPH. And since we've got 2 electrons there, right, it all works out. So the end product of the light reaction is ATP and NADPH. And where is this happening? In the thylakoid membrane. There we go. Bing. Okay. All right. So this is our major electron carrier to carry the energy of electrons, okay, to the Calvin cycle. So big picture here is that the thylakoid membrane converts light into ATP and NADPH. In the process, water is split so that oxygen gas is given off. So the part of the equation we've used is water and light. And now this is our intermediary carrier to take energy onto the Calvin cycle. So we're going to need ATP and NADPH for the Calvin cycle. So you're expected to understand kind of the big picture here. You are responsible for knowing that photosystem 2 comes first before photosystem 1. That photosystem 2 is P680 with the chlorophyll A. That photosystem 1 is P700 with chlorophyll B. Probably helps to dig out some animations of this process and watch it a few times so that it really gets in your head. This is not something you can learn right away. All right, so putting it all together, back again with this same picture. What's the big points? We've got two places where light comes in. Photosynthesis is undergonic. The electron transport chain is driven by light energy. All right, so sun comes in. 2 electrons come off of photosystem 2. Split water. Oxygen is given off. Okay, you get a bunch of protons creating a proton gradient. And the proton gradient drives ATP synthase to make some ATP. That ATP we'll use to make our sugar. Right? Sun hits photosystem 1. Okay, we still have 2 electrons to move on. Remember we talked about where those come from. All right, and those 2 electrons drive this process so that we can convert NADP into NADP plus into NADPH, this powerful electron carrier, which goes to carry energy to the Calvin cycle to make sugar. All right, and here's our summary slide. This is the one you should definitely pause and jot down if you've totally lost. Okay, the electron transport chain of photosynthesis uses light energy to produce NADPH and ATP to go to the Calvin cycle. Photosystem 2 absorbs light. An excited electron passes from chlorophyll A to the primary electron acceptor. Right, we have to replace that electron in chlorophyll. There's that powerful drive. And an enzyme extracts electrons excuse me, from water and supplies them to chlorophyll. So water splits and oxygen combines with another oxygen to form oxygen gas, which can be released to the atmosphere. And we breathe easier. So that's how we get oxygen in the atmosphere. So where did that O2 come from? Okay. This is how this was discovered by using a radioactive tracer called oxygen-18. This experiment is in your book, I believe. Maybe not. It's a kind of classic. This is how they figured out how the oxygen occurred before they knew all these details. I don't want to get into too much with it, especially if it's not in your book. This is super ridiculously long already. Okay. So the whole point of this experiment, I think it's somewhere in your book, but it's just to prove that the oxygen came from the H2O and not the CO2, because of course we have some oxygens floating around there, and if you didn't have evidence about the photosystems, you didn't know exactly where it came in, but it was an experiment to figure out that the oxygen for releasing this gas came from the water and not CO2. Okay, we do have non-cyclic photophosphorylation to go through, but this is a 48-minute video so far. We're going to end right here, and I'll make a short video of that.